In this series of conversations, we'll be discussing global sustainability with guests who bring a deep understanding of the environmental and cultural challenges facing our society and creative ideas on how to address them. I'm Ash Sweeting and welcome to the Ash Cloud. Today we are joined by Itzik Mizrahi, a microbial ecologist at Gurion University of the Negev. The Mizrahi lab studies the ecological and evolutionary forces that shape microbial communities in nature and specifically in gut environments. His work on the rumen microbiome investigates the ecology of the microbial community to understand cooperative and antagonistic interactions between species with the aim of improving food security and minimizing the environmental impacts of livestock production. The study of microbial ecology has many similarities to macroecology.
Microbial ecologists, such as myself, were asking the same exact questions as people that are actually studying macroorganisms, elephants, impalas, etc. So the general notion when you do that is to understand the individual importance of a single species within the ecosystem, how these species interact with other species, and how the cumulative web of interaction are translated to what we call the ecosystem function.
Like in our communities, behaviours change.
And in order to produce methane, exactly like our example with the grass, you will need more than one species. You will need one species that will produce hydrogen, and can also produce CO2, carbon dioxide, and you will need another species, at least another species that will take this hydrogen and carbon dioxide and will transform it to methane.
Context is everything and impacts decision making.
The context of a single organism that you would deem as a good guy, for example, would the context itself that it resides within could change this good guy to be a bad guy, meaning that in a different context it will be a bad guy. And it goes the same for consortia. So the function itself, the interactions themselves are extremely context dependent, as I see that. There is extremely, let's say, plastic. They have the ability to make decisions, microbes, and it's also context dependent. So let's say that this and the two are extremely interconnected. So let's say that you have a set of genes that would make you a bad guy. So it depends on what you decided to do, and you have another set of genes that will enable you to be a good guy. So in a certain conditions, not even the context of different microbes, you can express the genes that will allow you to be a good guy. In a different context or environment, this set of genes will make you a bad guy, exactly as we know from ourselves.
Complexity comes in many layers. That complexity is critical for both productivity and environmental impact.
We found that there are specific consortia, we call them ecosystem states, specific microbiomes within cows underneath the same exact diet. They are eating exactly the same diet, specific microbiomes that would be more beneficial for food security, because we need milk. And there would be other combinations that will be less efficient, will produce less metabolites that are could be transformed into milk by the cows and will be, for example, emitted to the atmosphere, like methane. Now, it's not that you have either one or the other. It's you have a specific combination that could have, for example, microbe 1 in both, but microbe 1 in a combination of 60 units of microbe 2 would behave differently than microbe 1 with 20 units of microbe 3. So the combination itself, it's extremely important. And one of the questions that we are now highly engaged with is how do we tip the scales between these two different what we call community states. Another layer of complexity that we published, I think, last year, is that when we look at the expression profile of the genes, which genes are actually being used by the microbes, which enzymes, we see that the same exact microbe in these two different community states will express different sets of genes. And these sets of genes allow us to actually predict the function in a way more accurate manner than actually looking at the combinations themselves. So we look at the genes that are being expressed by a single microbe in two different conditions, and we can say if these conditions are beneficial for food security, for example, if we get more milk from them, or more methane will be emitted. It's mostly connected to the way the microbes utilize hydrogen, but to many other pathways as well.
How do we hack microbial communications?
Microbes actually communicate. There are chemicals that are being used by microbes in order to communicate and to know how many of a single species they are, and microbe eavesdrop on other microbes and to know what they're actually talking about. But and this is sometimes called quorum sensing. But we're not talking about this. We're talking more about the transfer of chemicals from one microbe to the other. And then we're trying to hack into this metabolomic language, let's call it like this.
Community communications have impact.
This is where our ability to actually modify and modulate actually lies. So in terms of very simplistic terms, if we will increase the amount of lactate in the community, we know that specific microbes will utilize specific pathways that will allow them to utilize hydrogen, for example, better. And this will directly create a situation in which it will reduce the amount of methanogens in this community and therefore reduce the amount of methane, for example, because it's very deterministic, it's very repetitive. And we're trying to expand this to create a more complex communities to better enable us to predict and to what we call engineer this type of communities.
The natural variation in methane emissions is huge.
In the rumenomics study, we had cows that would emit 100 grams of methane per day, and we had 500 grams per day from a different cow in the same herd. So it's 5 times more within the same herd. So you can reduce it
the same
5
diet
times.
And the same everything else.
Exactly.
The timing of modification is critical.
Kind of breathing towards lower methane emissions is one direction. Another direction that I highly believe in is the ecology of ruminants. We've shown previously that you can actually modify the assembly process of the microbiome if you intervene early on at the beginning.
Itzik, thank you very much for joining me today.
It's a pleasure to be here, Ash. Thank you for having me.
My pleasure. You and your lab, you study the ecology of microbes, these tiny little organisms that are ubiquitous all across the world in every single system. Can you start by telling us a bit about the ecology of microbes, how they interact, and then especially how they contribute to some global issues like ruminant methane production?
Yeah. So essentially, if I would have answers to all of these questions, I would maybe retire. But I can maybe better describe how do we ask these questions and why, maybe. I'll start by explaining the notion of microbial ecology as I understand it. So essentially, microbial ecologists such as myself were asking the same exact questions as people that are actually studying macroorganisms, elephants, impalas, etc. So the general notion when you do that is to understand the individual importance of a single species within the ecosystem, how these species interact with other species, and how the cumulative web of interaction are translated to what we call the ecosystem function. So if I will take my metaphor for macroorganisms and I will talk about the savanna, and we have the ones that eat the grass. So if we, so this is a type of trophic, and we omit one of them, the other will essentially deteriorate in numbers, but more importantly, the ecosystem function will be affected. If we will omit the lions, we will have less grass in this ecosystem. And if we omit the impalas, we will end up with very high amounts of lions that will eventually die. So this type of interaction could be affecting both each of the individual species, but they also affect the ecosystem function, because when you have a lot of lions without impalas, you have a lot of grass, and the other way around, when you have a lot of impalas without the lions, you will end up with no grass. So the ecosystem will be affected. In the same manner, when we study microbial communities, they also have trophic interactions and other types of interactions. And unlike what we do in the savanna, where we actually see the output of the interactions, here we can only measure the effect on the environment or the concentrations of specific chemicals. And we can measure direct interactions between two microorganisms or a group of microorganisms. And we can also infer this type of interactions by either omitting or looking at an ecosystem in which one of the species is not there. And we're doing it with what we call multi-omic approaches, but also very classical approaches like growing these organisms and looking at the actual effect, direct effect on each other, or even indirect effects when they grow together. And when we're using the multi-omics approaches, we are looking at them as a community. We're looking at the overall genomes in a process that we call, or in an approach that we call metagenomics, or the overall proteome, the genes that they express, or the metabolome, which for me is very important because we are actually looking at the actual chemicals. Now I'll connect it to methane emissions, productivity, as you just asked. So if we look at the ecosystem function of a microbial community that reside within ruminants' guts, the ecosystem function could be measured from different perspectives. One of them could be the amount of methane that will be emitted to the atmosphere. And in order to produce methane, exactly like our example with the grass, you will need more than one species. You will need one species that will produce hydrogen and can also produce CO2, carbon dioxide. And you will need another species, at least another species that will take this hydrogen and carbon dioxide and will transform it to methane, which essentially it's a reduction reaction of CO2 into methane. And in this context, you have a community of two microorganisms, and of course it could be way more. One that will produce the CO2, another one the hydrogen, different amount of hydrogen, and the methanogen that will convert the CO2 and hydrogen into methane. So once you have more than one organism, this is a community of two that I'm just describing, you can measure the ecosystem function, then you can think about what happens to the ecosystem function when I reduce or increase the amount of one of these organisms. What will happen to the amount of methane being emitted? From the other point of view, the amount of energy that is being converted to things that the animals can use, like propionate, acetate, butyrate, is also an ecosystem function that is directly being translated by a cow, for example, into energy that it can use, could also be measured. So therefore, and in this context, it's a way more complex interaction between multiple different organisms. In a second, we'll discuss about that, in which we can actually measure. And what we end up with is essentially very complex trophic interactions between multiple different species that might end up with more energy to the animals or less energy and more methane emitted to the atmosphere. We didn't discuss the physiology that allows these processes to happen within animals, but the end product would be either high methane or a lot of chemicals that the animals can actually use in the case of cows.
Thank you very much. I love the analogy you used in terms of the savanna and the impala and the lions. I think that's great. And that explains things very clearly about how one thing changes and there's all these flow and effects. Obviously, the analogy of the savannah is quite simple with two species. How complex is it within I guess both the rumen microbiome, but also other microbiomes that you look at?
Okay, so maybe I'll start by expanding my answer to the previous question and framing this answer and explaining how more or less the rumen microbiome works and how does it interact with the actual animal, which is a ruminant. In our case, it's mostly cows and sheep. So these animals, somewhere in evolution, enlarged their esophagus into a large compartment called rumen. And within this compartment, they essentially grow microbes on the grass that they ingest. And once the grass is getting into this chamber that we call the rumen, it meets a very complex community of microorganisms. And this community essentially degrades the plant material, starting with the polymers like cellulose, hemicellulose, starch, into simple sugars, the monomers, dimers, trimers, that are then being utilized and fermented into short-chain fatty acids through a very trophic-like process by multiple different species. Or being converted into methane, again, by kind of a cascade of reactions by different species. So we're talking about, in the case of cows, hundreds of different species. There are other organisms where they have more simple microbiome, less complex, with less amount of species, less rich. And there are others that have similar numbers, but ruminants are have one of the most complex microbiomes. And in our work, as we just described, we're studying direct interactions, like eating the impalas by the lions, or the impalas eating the grass, or indirect interactions, how the grass is being affected by the presence or absence of lions, because they do not interact with the grass directly. And so how to study that? Or how complex is their study of these very complex organisms that we cannot actually see. When we do that, we rely on the pillars that were set by ecologists like Hutchinson and others that allowed us to connect species interactions and ecosystem structure to associations. At first, what does it mean? It means that, as I described before, in order to infer a function or an interaction between two organisms, I do not necessarily need to measure a direct effect between one organism and another, or between one organism and the actual function. So if we go back to the things that we can actually see and touch, so let's say that we're looking at multiple different islands and organisms on these islands, and if I don't see a certain organism, actually see macroorganism on a specific island, and I see a depletion of a function, I can assume by association that this organism is performing this specific function. Or if I look for co-occurrence between two birds on a specific island, and I see that whenever I see one, I would see the other, I would presume that they have some kind of a positive interaction, and vice versa. If I see one, I would necessarily not see the other. I would presume a negative interaction. And it goes the same for the genes that they carry, and also to the chemicals that they express. This is an association study, and the study of microbiomes started like this, and it's still using these tools, but I see them specifically in my research as hypothesis generating tools. And once we discovered or we identified such associations, or suspected associations or interactions between species or a specific species and a certain function, I would usually go for a more causal approach in which I will actually measure the interaction between one species and the other, putting both of them inside a tube or both species inside a sheep. We have germ-free sheep facility in our group, or a complex microbiome inside the sheep, and then try to infer how the ecosystem of multiple different species would look like, how the ecosystem function would look like, how the interaction between the species would directly look like by omitting or depleting specific species or adding other species. So it's very complex. It requires both, as we said, multi-omics approaches that allow us to generate this hypothesis, and then a lot of robust classical microbiology that is combined with a lot of techniques, like we are now using robotic systems to actually allow us to culture and play with combinations of different microbial species inside tubes.
Thinking about what you've just said, well, firstly, there's quite a few questions that come up, but the first one is that go to so many parts of our society, whether it's the film industry or whatever. We kind of like to have good guys and bad guys, and there's the one team and the other team, and it's like, well, let's support the good guys and they'll take on the bad guys. But it seems to be much less, I guess, linear than that. And it's all about a slight push in one direction will have multiple effects in multiple different places that won't necessarily, obviously, there's that, Newton's law of thermodynamics, that each reaction has an equal opposite reaction, but the reaction is not necessarily where you'd expect it, or linear between, the good guys or the bad guys. That must make it one fascinating to look at, but also something that takes a lot of thinking to actually try and get your head around that.
Yeah, well put. Maybe even I would add to this 2 different layers of complexity. Yes, it is not linear. The reason that we are sometimes using what we call network analysis in order to detect such interactions that are not linear, not directly direct. We can look at indirect interactions. And more than that, the context of a single organism that you would deem as a good guy, for example, would the context itself that it resides within could change this good guy to be a bad guy, meaning that in a different context, it will be a bad guy. And it goes the same for consortia. So the function itself, the interactions themselves are extremely context dependent, as I see that. There is extremely, let's say, plastic. So you would find it's exactly like when you go into a bar, you can meet somebody that could be amazing to talk with, but in a different context, you would think on this person as an idiot. It only depends on the context. So this is one level of complexity. It's highly context dependent, how you would deem a microbe, whether it's good for the ecosystem function or bad for the ecosystem function, for example, one thing. And I will maybe translate it to something more like something that we can actually wrap our head around, and it won't be abstract in a second. The second level of complexity, and this is embedded in several studies that we did recently, and I can talk about them in more details in a second. They have the ability to make decisions, microbes, and it's also context dependent. So let's say that this, and the two are extremely interconnected. So let's say that you have a set of genes that would make you a bad guy. So it depends on what you decided to do. And you have another set of genes that will enable you to be a good guy. So in a certain conditions, not even the context of different microbes, you can express the genes that will allow you to be a good guy. In a different context or environment, this set of genes will make you a bad guy, exactly as we know from ourselves. So there is what we call functional plasticity within the microbial world that makes it a bit more complex. Now let's wrap our heads around what I just said with regard to methane emissions maybe and productivity of animals. So we found that, and I think Sharon once in one of your podcasts described that, we found that there are specific consortia, we call them ecosystem states, specific microbiomes within cows underneath the same exact diet. They're eating exactly the same diet, specific microbiomes that would be more beneficial for food security, because we need milk. And there would be other combinations that will be less efficient, will produce less metabolites that are could be transformed into milk by the cows and will be, for example, emitted to the atmosphere, like methane. Now, it's not that you have either one or the other. It's you have a specific combination that could have, for example, microbe 1 in both, but microbe 1 in a combination of 60 units of microbe 2 would behave differently than microbe 1 with 20 units of microbe 3. So the combination itself, it's extremely important. And one of the questions that we are now highly engaged with is how do we tip the scales between these two different what we call community states. Another layer of complexity that we published, I think, last year, is that when we look at the expression profile of the genes, which genes are actually being used by the microbes, which enzymes, we see that the same exact microbe in these two different community states will express different sets of genes. And these sets of genes allow us to actually predict the function in a way more accurate manner than actually looking at the combinations themselves. So we look at the genes that are being expressed by a single microbe in two different conditions, and we can say if these conditions are beneficial for food security, for example, if we get more milk from them, or more methane will be emitted. It's mostly connected to the way the microbes utilize hydrogen, but to many other pathways as well. So I don't know if I answered your question properly, Ash.
No, thank you. Thank you. That was wonderful. Am I in many ways this leads me to think that in some ways it's a bit like a communications exercise. You need to somehow communicate to the different communities of microbes that you would like to do metabolic pathway 1 rather than metabolic pathway 2, or it's more about rather I think of it like they're communicating and we need to work out how we can influence their communication.
Exactly.
What do we know about how they communicate and the opportunities to actually or what do we need, or what more do we need to know if we don't know terribly much about it, but in terms of how we can influence that communication?
Okay, cool. Actually, this is what our research now is mainly focused on. So when we say communication, I just want, before we talk about it, just to explain that microbes actually communicate. There are chemicals that are being used by microbes in order to communicate and to know how many of a single species they are, and microbe eavesdrop on other microbes to know what they're actually talking about. But and this is sometimes called quorum sensing. But we're not talking about this. We're talking more about the transfer of chemicals from one microbe to the other. And then we're trying to hack into this metabolomic language, let's call it like this. So we know that there are community states, as we just described. We know that they express specific genes as a function of the context of other microbes that reside within this community, and as a function of the conditions, the physical conditions inside this community. Now, what we are trying to do now is to, as you just said, to understand or to get a good understanding for the patterns and the level of determinism, the level of, let's say, our ability to predict it, how constant are these patterns across different microbes in specific states. What does it mean? For example, I'm speaking to Ash. Ash is a very nice person. I know that when I will say good morning, he will reply with a very nice words to me. And then I see another person, which I know that whenever I will say good morning, he or she will not answer. So this, and it will happen day after day after day. So this is a very deterministic behavior that when I know how I start, I will know where I end. So once I know if I use a certain chemical, these genes will be expressed by this microbe, or if I will have this microbe in the community, this will create a situation of other microbes that will interact with this specific microbe in a deterministic manner. And this is what we're doing. This is where our ability to actually modify and modulate actually lies. So in terms of very simplistic terms, if we will increase the amount of lactate in the community, we know that specific microbes will utilize specific pathways that will allow them to utilize hydrogen, for example, better. And this will directly create a situation in which it will reduce the amount of methanogens in this community and therefore reduce the amount of methane, for example, because it's very deterministic, it's very repetitive. And we're trying to expand this to create a more complex communities to better enable us to predict and to what we call engineer this type of communities.
I think maybe thinking in awe about early rumen microbiologists that didn't have the luxury of using electronic computer systems and AI and machine learning, or the tools, or genomics, the tools that we have today to understand all this. How important is that technology, and is there things that you would like to see, or other technologies you'd love to see pointed at this problem?
Yeah, so that's a great question. First, I want to commend the people that I call prophets, people like Hungate. These people actually predicted a lot of the things that we're doing nowadays. I will give two examples in a second. Using AI, let's start from the end. We started doing that at, I think, 2012. And this proved to be very, very, very strong approaches that allowed us to understand the patterns without really understanding them. So this is the good and bad thing when we compare to what they did in the old days. We can actually crunch tremendous amount of data, and we kind of tweak these algorithms to allow us to do that and to get a prediction level saying, okay, if you have this ecosystem, this will be the function. Looking at multiple different species, this is what the AI gives us. What it doesn't give us, sometimes the AI are kind of black boxes, and we're invested a lot on their decision making. How do they actually understand? We are teaching them, or they are learning from our data, and they have this secret how they decided on something. Let it be tree decision making, and neural networks are even more complex, where we try to understand how they actually made the decision. And this gives us a lot of let's say fundamental understanding with regard to the ecosystems themselves. So from one hand, they allow us to crunch a lot of data and make decisions and make predictions. And from the other hand, we're lacking what the prophets like Hungate had, where there is a very clear understanding what causes what and what affects and towards which directions they actually affect. So the notions of hydrogen utilization that we're now talking about, and the notion of how species interact within the ecosystem, these people actually fully understood that. We're now expanding their knowledge. This is what I feel that I'm doing. And we're still lacking a lot of, in order to fully use AI to its maximum extent and to get what it actually can give us, we need to have way more data than what we actually have today. We're using types of algorithms that are designed to deal with sparse data, as we call it, less amounts of data. And when we have enough data, I think we can use more robust algorithms that are being used, for example, for image processing in a way that will enable us to predict way better, exactly as Facebook works and other algorithms that that can actually identify pictures. So to make a long story short, the deep biological thinking and the hypothesis-driven thinking is extremely important in order to interpret how the decision-making of AI machines or algorithms are working. And in order for these methods to be fully let's say, beneficial for us, we need to expand our data generation methodologies.
What areas of, in terms of expanded data, what areas is it across the board, or are there specifics in that regard that you're looking at?
Yeah, across the board. This is the very plain answer to you, but I will just give some examples. So we were part of a very, very big study in a project called Rumenomics, in which we, in this project, 1,000 cows were sampled for their microbiome, their methane emissions, short-chain fatty acids. It was led by three amazing scientists. One of them I had the privilege to actually work very close with. His name is John Wallace. He's one of my favorites. He's a great guy. So in this data set, we had metabolomics, microbiomes, and methane emissions of multiple different cows, 1,000, and the diets themselves. Phil Grimsby helped us a lot with the actual understanding of the physiology. It sounds like a lot, 1,000 cows, yes? But it's not a lot in terms of machine learning. Like, in order to fully use and grasp the data, I would have been glad to have 10,000 cows, for example. But this is enough for us. It was enough for us to actually predict. With regard, for example, for metabolomics and how metabolites are being utilized by microbes, produced by microbes, and how they use them in order to interact, hacking their communication, as you defined it before, the data is really, really, really poor. We have not enough. And more than that, in order to play with different combinations of communities, for now, we don't have enough species that cover the overall microbiome composition. And our lab, and I know Sharon and other labs, are now involved in isolating multiple different microbes that will allow us to cover this diversity. And we collaborate as a community on this, because this is extremely important if we want to recapitulate the ecosystem and actually direct it towards a specific function.
So going back to your Facebook analogy, I'm not quite sure of the exact numbers, but from my understanding, it's the number of humans that use Facebook, at least, occasionally, is more in the billions than in the millions, which is orders of magnitude greater than the 1,000
exactly
cows that you. So it's kind of that scale where the real power comes from.
Exactly. And but people are working on ways to actually teach these algorithms with lower amounts of data, creating spurious noise, as we call it, like multiplying each sample to be way higher than it actually is, in order to create a situation that they can learn better. So I think that in the coming few years, both our ability to expand the data and the ability to utilize less amount of data, both will be, I think, better, because this is a real need of the field.
I'm not going to ask you to predict the future, because I know that is way beyond both of our pay grades. However, where do you see opportunities for hope? When you think about this, which you do deeply on a daily basis, what might you see as a potential solution or potential avenue where, say, methane emissions are addressed in ruminants in 5 or 10 or 20 years' time?
Yeah, so I can tell you what are the current approaches, what is under researched, and where do I see let's say, the bright sides of what we already did. So I think in terms of where we want to go, we more or less, at least in my group, and I hear other people talking about this, more or less we know what would be the direction. How to get there, I mean, like in terms of community composition that is better and might reduce methane emissions tremendously. Just to get a grasp of it, in the rumenomics study, we had cows that would emit 100 grams of methane per day, and we had 500 grams per day from a different cow in the same herd. So it's 5 times more within the same herd. So you can
And that's
reduce
on
it
the same
5
diet and the
times.
same everything else.
Exactly. So this is crazy because they were never actually individually measured. Um, um, and we know what are the community compositions that are beneficial or not. So how to do that better? There are several approaches. In this study, we kind of connected microbes to the cow's genome, and also people are directly trying to breed cows for lower methane emissions. I know that it affects their productivity. I think that there is, once you breed to lower methane emissions, you might end up with different community composition and different physiology. I think that people should take the microbiome into account when they're doing that, and also, the effect on productivity. So kind of breeding towards lower methane emissions is one direction. Another direction that I highly believe in is the ecology of ruminants. We've shown previously that you can actually modify the assembly process of the microbiome if you intervene early on at the beginning. Others, but it's controversial, others did something, things that are different with diets. And I think that the moment of intervening is extremely important. If it's being done just after birth, we might be successful in modulating the ecosystem function a bit. And this is what we need, because cows, at the end of the day, the majority of the microbiome is identical. We can change the flexible parts of it, and which I think are extremely important with regard to methane emissions. So if we intervene early on at the beginning with specific components, we might end up with lower methane emissions. So this is another direction. A totally different direction is maybe trying to use feed additives, as you heard about, and few are working and are safe. I don't want to use this stage to actually name them, but some of them we know that are actually working. They at the same time they cause some alternations in the physiology of the rumen. For example, increase in the amount of hydrogen. So I think that intervention at this level with specific microbial communities could be extremely important, because this could increase productivity. For example, utilizing the high amounts of hydrogen towards production of propionate, which is extremely important in the gluconeogenesis process inside the liver of cows after it's being absorbed. There are other feed additives that are being tried now, and there are some discussions around these notions because they are not endogenic to the cows and they were never tested. So I think that the solution is always, as a scientist, I always believe to go with towards a solution with different approaches. So I would go for feed additives, the ecology of the microbial communities, and maybe selecting for specific cows with lower methane emissions could be a kind of a combined approach to reduce methane emissions.
Do you see opportunities for the next generation biotech technologies like synthetic bio or gene editing of microbes, or other technologies to come in, and we'll see something and then change the biology? Do you see that as an exciting path forward, or is that something that you see as otherwise?
So I will start by saying that innovation in general is cool. So I'm a groupie of innovation. And as I said, we should try everything. Judging by the past, by modulating one genome or two genomes using CRISPR-Cas or whatever you can think of, and introducing it to a very stable and, let's say, very complex ecosystem, could sometimes be not as beneficial as using nature itself. So there were trials of introducing a specific gene that will detoxify a specific component in the rumen. And it proved to be successful to some extent, but it could have been solved, I would say, in a way easier manner if you can actually study nature. So, but you never know where this innovation, gene editing, etc., could end up with. So I would definitely try this in synthetic microbial communities, see if I can integrate that. But my go-to approach would be to look at the variability existing in nature in these microbial communities and trying to enrich towards a specific microbial community composition that already exists in nature, and not trying to introduce something new.
If
But
I
I would never.
if I understand what
Sorry.
you're saying correctly, I think that's if you're targeting something as singular as a gene editing or a CRISPR approach or something like that, that goes back to assuming that works in this one-to-one type linear good guy bad guy situation, whereas it's much more difficult when there's so much variability and there's someone is a good guy sometimes, a medium guy, and other times a bad guy, and that all could change multiple times within a very short period of time. And so you need the nature approach works because it accounts for that complexity, whereas if we take that linear mentality, we'll end up in a rabbit hole and everything else will be going on around us. And is that a fair way of thinking about it?
Yeah, except of the rabbit hole at the end. So I would say you would never know what works, and I would never, like, it's just a belief that I have, that I wouldn't say something not to work or forecast that it wouldn't work because I didn't try it. But what I'm trying to say is that biology is a science that deals with multidimensional parameters all the time. And we have hard time understanding how each one of them affects the overall ecosystem and how they interact pairwise in a very complex system. So my general approach would be to take a top-down approach, meaning to understand how nature works, and then maybe to work bottom-up. It could be using gene editing, but then you're increasing sometimes complexity or introducing new innovation that you do not know how it will affect the overall ecosystem. So I'm not against it necessarily, and not like, definitely not. I'm just saying if it's being done in a rational manner, and with maybe a direct going back to the good guy, bad guy, after you pinpointed the bad guy in every context that you would look at, this would be the bad guy, you might end up with a CRISPR or a gene editing approach to affect it, because it would have a very high impact on the overall ecosystem. So maybe this is a more complex way of saying that I would start with trying to understand nature, then modifying it one by one.
Excellent. Well, I commend that approach, and I guess as an ecologist, that's exactly what you do, is you try and understand nature and all those interactions, and you've chosen, it's much more difficult and challenging to look at the ecology of the microbiome than those impalas and lions wandering around the savannah.
I'm not sure that if you would ask the ecologists that are doing it in the savannas, they would agree, but I guess it's highly challenging as well.
Well, I guess the ecologists in the savannah also have to deal with the microbiome across that savannah, how that moves between the impala and the soil and the plants and the lions and all that sort of stuff as well.
Exactly, and the lions can eat them, So there is another layer of complexity for you.
So a slight change of tack, if that's okay with you. And you're Israeli, you live in Tel Aviv, and
True.
you guys have created it's a desert, there's not a lot of water, and you created some of the most water efficient, greatest sort of ag tech products as well. And you understand food security and how that relates to your, I guess, your national security. What do you living in that environment, what do you have you can share with us about those relationships?
Yeah. Okay. Thank you for this question. So so I grew up and I was born in Tel Aviv, and until 3 years ago, I was living in Tel Aviv. Part of me thinking about these concepts is directly connected to the fact that Israel is a country with very low amount of resources. I recently moved to the actual desert. Now I'm living in a village in the middle of the desert. It's called Lehavim, very close to the Ben Gurion University where I moved to. It's again a university at the south part of Israel where you have a desert. So maybe the main thing that you know, and you kind of, it's kind of your part of your essence living in these areas, is that scarce resources enforce you to be innovative. And more than that, when you don't have any other choice, you need to be innovative and you need to find a solution to the problem because, you know, your life, your existence depends on it. And I think part of what just happens to the world is very similar to how Israel lives for years now, because in the essence of global warming is that we don't have any other choice than actually stopping it if we still want to live in this planet. So if I'm taking the notions in Israel and the way, even the way we grow cows here and the way we utilize water, about 60-70% of the water utilization in Israel is not coming from, it's coming from desalination or recycling. And we had to do that very early on. And now the world is doing that as well. So taking these notions is that once we have a problem, there is a way to solve it. And there is a way to get into the end goal of trying, for example, in our case, to modulate the rumen microbiome. We just need to be innovative enough. And I think that this era, although it's very problematic in this context, that we need to feed a lot of people on this planet. We're about now between 7 to 8 billion. We don't have enough resources. And at the same time, this process, this intensive farming is affecting the globe in a very negative manner. So maybe this era that we're living in is very alarming from this context, but I think it's a stepping stone or an inflection point for humanity to accelerate its way of thinking towards a more sustainable thinking on living on this planet. So from this perspective, I'm very glad to live in this era. And I'm sure, and I'm quite sure that we will find a solution. And I'm quite sure that this opportunity, although it's a challenge, this opportunity that our generation is facing is a bless in this context. In this context, I'm very excited, let's say.
Wonderful. Is there anything else that you think we should discuss or you'd like to add that we haven't talked about so far?
I just want to say that I know other communities. I just want to say that our community, the rumen microbiome community, we know each other. And we have the ability, like, for example, I just mentioned the ability to, the need to isolate a lot of microbes from the rumen microbiome. We have the ability to communicate with each other, and we have the ability, and we're doing that. And this is really exciting in this community, to direct the overall community towards a certain direction and to collaborate in a way that would be very, let's say, beneficial and efficient. And actually, with this, I'm very, very happy that I'm involved in the ecology or the microbial ecology of the rumen. Like, I have amazing colleagues that I'm very happy always to see, but also to work with, because we're actually trying to solve very, very big problems for the world. So from this perspective, it's a huge privilege.
Excellent. Itzik, it's been an absolute pleasure speaking to you. Thank you very much for taking the time.
This was a real pleasure, Ash, and thank you for having me.
You've been listening to The Ash Cloud with me, Ash Sweeting, in conversation with Itzik Mizrahi, recorded in California in May 2023.